This is Robert Sapolsky, and you're listening to Father-Offspring Interviews.
Howdy, folks. Welcome back to Father-Offspring Interviews. First, Dia asks, could you please let us know about mirror cells?
So, we're all worried about lots of things. About getting hantavirus or Ebola virus or flesh-eating bacteria, or how to correctly pronounce cyclosporiasis, or is it sporiosis, let alone if somebody is putting the Leviosa or Leviosa spell on you. All these things to worry about. This question brings up something so obscure and out of left field, which turns out to be a hell of a lot scarier than all of those put together, and which in about 30 years or so may spell the end of life on Earth. Mirror molecules. So we go back to when the Earth was young, before there was any life, there were any cells, there were any complex molecules. We just had the proverbial primordial soup filled with incredibly simple little chemical elements floating around there. And one of the most important things to come out of like the invention of life is because of the chemical nature of these very, very simple chemical structures, if they would bump into each other, they would occasionally bind together. And then they would bump into some more and bump into some more and play that out over, I don't know, a billion years or so. And gradually, this is how you start getting complex molecules, the pathway towards life. So that's one scenario. In the 50s, there was this incredibly famous experiment by Harold Urey, University of Chicago, Nobel laureate chemist, and his faithful sidekick Miller, who was his grad student, who hasn't been heard from since. But the famed Urey-Miller experiment, where they took a primordial soup and filled it with incredibly simple, primitive elements and passed electricity through it, like lightning strikes. And they showed this greatly accelerated random bumping of these simple thingies together to begin to form more complex thingies. Aha, that's how life started. What people have learned since then is electricity is nice to facilitate that, but molecules, very simple chemicals, elements have a property of being self-organized in their complexity. So that's a pathway by which life started. Okay, so we are going to take a very simple version of that. We are going to have a primordial soup here as shown that consists of only two thingies. And they're randomly floating around, two versions of them. And what you see is each one is sitting by itself, all on its own. And the jargon would be it's a monomer. And by the bumping together scenario I just outlined, if two of them bump together, they form a bond, a stable bond. And two of them we now call a dimer. So there's a number of different versions of dimers that you can get. As shown here, simple thingy A might bind to simple thingy A, and what we have is a homodimer. It's pure A. Or simple thingy B to B, another type of homodimer. Where things get interesting is when an A and a B bump together and you have a heterodimer. There's two possible configurations of that. Let's call A the one on the left. Let's focus on A on the left, whose left hand is holding hands with the other thingy. So we're going to call it the left-handed conformation. And it can get flipped the other way with what we would now call the right-handed conformation. And it turns out that every single molecule on Earth has the potential to have a mirror image of itself. This is a property in chemistry called chirality. If you hold something up like a perfect sphere to a mirror and it's completely superimposable, this is something that does not have chirality. If you hold AB up to a mirror, and BA is what you're seeing reflected in it, AB cannot be superimposed on BA. BA is a chiral molecule. It has left-handedness and right-handedness. Okay, so it turns out all of the complex molecules on Earth can come together in chiral ways, be left-handed or right-handed. So you've got all these simple chemical structures, when they come together, can form left-handed chiral versions of themselves, right-handed ones. And just to show what we're dealing with here, if you have a mixture of both the left-handed version of A and B, AB, and the right-handed BA, you have what's called a racemic mixture, both together, A and B, both together, left-handed and right-handed. So that's the way molecules come together in the universe. They can have a left orientation or a right orientation with this chirality business. But then comes a staggeringly implausible finding, which is every living organism on Earth, all of the molecules that make it up are monochiral. Every single molecule is just one of the two potential hands. Okay, 30-second footnotes here. If you are already lost, don't listen to a word I say now, but it is not the case in living organisms that every single molecule is left-handed or every single one is right-handed. One class of molecules, like amino acids, will have the same handedness every time you find them. Another class will have handedness perhaps in a different direction. Main thing is living biochemical systems, all the molecules in there are monochiral. How'd that happen? We've got a world with both left and right-handed versions of molecules. When you throw them together randomly, A bumps into B, B bumps into A. How did life wind up having only one-handedness, having only one hand for every biochemical molecule out there that you find in living things? And when you think about it, there's actually a certain logic to it. So suppose you got AB and BA, you got a racemic solution, a mixture of both, this heterochiral solution, and you're building a body on it. And it turns out if you were able to break the bond between A and B, you're going to get energy. Whoa, what a terrific thing. Let's figure out how to develop an enzyme that could clip A and B. But it turns out you're going to need the chiral version, the mirror version of that enzyme to clip B and A. If you're going to have living organisms be heterochiral, if they're going to have left and right versions, you're going to have twice as many enzymes, twice as many receptors. Everything's got to come in a left-handed flavor and a right-handed flavor. And what you see is organisms sort of picked going with only one of them and saved half their energy, Darwinian selection. And now we have this bizarrity that all of life is monochiral. So you make synthetic molecules in a lab and you just throw stuff in and you wind up with a mixture of left-handed, right-handed. And when you're doing drug development, typically the one-handed version of it is effective and the other is totally neutral. So you make this racemic mixture of both, and basically only half of the molecules, the one with the correct handedness, goes in there, has the effect that you want out of this drug, and the other is completely neutral. Except it turns out the mirror image of some drug that was developed in the 1960s had an effect of its own. And this is one of the disasters in medical history. This was the development of a medicine that suppressed morning sickness in pregnant women, a drug called thalidomide, and it was put in a mixture, left-handed, right-handed. Left-handed was the version that suppressed morning sickness, and right-handed is totally neutral. No, this turned out to be the staggeringly rare case where the right-handed version of thalidomide had its own activity. What did it do? It caused massive birth defects, a nightmare in the early 60s. Kids born like this was the thalidomide disaster. This had to do with the fact that amid our world of living organisms being monochiral, every now and then the mirror version of some drug can be active. Okay, so we now have this issue of, can you construct a mirror version of some? Can you take a left-handed molecule, something you know and love, like a molecule of hemoglobin or a stretch of DNA or something, and can you do the chemistry to be able to make the mirror image? Do it in a lab and just throw stuff in, and you're going to get a mixture of both. And then it's a very challenging task to degrade all of the stuff from one hand, and now you only have the other version. In theory, if you can get enough of the building blocks of molecules that make up a cell, and in each case, make sure that type of molecule is the opposite version of what you normally see in us, you can get a mirror cell, a cell that is the chiral opposite of the cells that make up all of life. Okay, before we get ahead of ourselves with making mirror cells, how do you even make a cell in a lab? That seems like a non-trivial challenge, and that's been the holy grail for a long time. And interestingly, about a month ago, there was a massive breakthrough. The report from a lab of Kate Adamala at the University of Minnesota that had made the first synthetic cell. Not a mirror one, that's down the pike there, but actually made a synthetic cell, put together all the building blocks needed, and this thing self-assembled into a cell, and it kind of does amazing stuff. This thing that they called a spud cell, it can grow, it can divide, it can eat stuff, it can compete with other copies of itself. Whoa, that's terrifying. And what the scientists did was a sensible sort of thing when constructing something like this, which is you leave out some key nutrient, some key constituent that the cell cannot make on its own. It's not going anywhere outside the lab unless you dribble some of that missing stuff into their broth there. They're not going to survive. That's the safety net that's done with these sorts of things. So we now have these spud cells that have been reported. And of course, there's a huge, magnificent debate now as to whether these things are really alive, or are they lifey, or are they life-ish? But this has been a huge breakthrough in the last few months. And what that suggests is 10, 20, 30 years down the line, are the estimates, people are going to be able to make a synthetic cell that's a mirror cell, is the chiral opposite of what that molecule would be in us. And this is unbelievably dangerous. This is the thing that could be the end of life. How come? Okay, let's look at how your immune system works. Some invasive pathogen gets into you, and what immune cells are good at is grabbing some molecule off of this invasive thing, some protein on the surface of this bacteria. The immune system, surveillance cells, snatches up that piece of that alien molecule. And what it does now is go to other immune cells. It chops up this piece of alien molecule into tiny little pieces, and each one of those pieces is now presented to one type of immune cell and another, and this immune cell starts making antibodies like crazy against that fragment. This one's making antibodies. You now make a massive amount of antibodies. You run this whole elegant competition to see which of the antibodies are best at recognizing this alien molecule and not recognizing some important part of you. That's great. That's how your immune system deals with this. What's the implication here? As we went over before, you may have a very, very effective armamentarium of enzymes to break down proteins, molecules from any known invasive organism out there. But if it's a mirror cell, there's no mirror enzyme that exists to break it down. You can't fragment it to make antibodies. The body can't mount an immune defense against these things. And in theory, these are going to be unstoppable pathogens. Okay, this is mighty obscure and science fiction enough that recently a collection of 38 scientists, including Kate Adamala, the leader of that synthetic cell study, 2 Nobel laureates, calling for an Asilomar conference about mirror cells. Asilomar conference. Asilomar is this great place down the coast of the Pacific where there's a conference center. And very famously in 1975, all of the leading molecular genetic engineers, just being born as a discipline, got together in Asilomar and had one of the most impressive things ever. They sat there for days and figured out what exactly our experiments were willing to do, were not willing to do with this engineered life stuff. And the Asilomar conference was this landmark for scientists self-policing. And in the last couple of years, people have been calling like crazy for an Asilomar conference about AI with all of the AI moguls. And that sure isn't happening yet because all of them are very interested perhaps in the financial bottom line. In any case, recently, a bunch of these synthetic life chemists have come together, and 38 of them issued a call for an equivalent of an Asilomar conference for mirror life, to have some sort of decision as to what's going to be too dangerous to do. Already, there's various funding sources that are saying they are not going to fund research on making mirror molecules. This is so obscure, and this could come out of left field and be really consequential. So great question about those mirror cells.
And here I thought that infected lettuce was going to be our downfall. All right, we figured after that slight downer, we'd shift to a more joyful subject. So now Hans from Sweden says, there have been a lot of reports in the news recently about similarities between how humans and other apes laugh. How do we know that this isn't just anthropomorphizing?
Oh, what a killjoy question. Yes, there was a recent publication that got a lot of coverage in the press about the similarities in laughter across all the apes, including us. What's up with that? And the questioner brings up the inevitable question, which is, are chimps and gorillas actually laughing, or are we being victims of anthropomorphism? Anthropomorphism, taking some human specific trait and projecting it onto some other species. Oh, look, the 2 hummingbirds love each other. Oh, look, your pet, your dog loves you. Look how it just projecting on. And that's one of the most deflating things out there, to say that some cool observation where some other species is doing something that seems to be on a continuum with us, you're just anthropomorphizing. So how do you recognize it for that? How do you tell if other animals actually laugh, if other apes do? And this was a career's worth of work by this Estonian-American scientist, Jaak Pansep, who died a few years ago. And he came up with the name for this whole field of affective neuroscience, the neurobiology of emotion. And he wanted to study emotion in a range of species. Aha, you're just anthropomorphizing, seeing emotion in a lab rat, emotion in a chimp. He was mocked, especially by B.F. Skinner, no surprise. And in his later years, he started pioneering the study of laughter in other species. All right, let's start simple. Laughter in other apes, chimps, bonobos, gorillas, orangs, gibbons, siamangs. What you see is a type of vocalization that seems remarkably similar to human laughter. Apes will rapidly emit air. I'm going to do a very lame version of it now. It sounds not at all like that, but it's a rhythmic, broken up exhalation pattern, a lot like what human laughing is about. Okay, so how do you figure out if this is laughing, if this is not just anthropomorphizing? First thing, do you see other apes making these sorts of vocalizations in the same contexts that humans do? Yes, indeed. When two adolescent chimps are playing, when they're wrestling, when they're tickling each other. Chimps love to tickle each other. The same body parts, the same part of their body that's ticklish, and they love doing that. And when they're tickling each other, when they're wrestling, when they're rolling around, when they're doing that, they make these laughter-like vocalizations. What this new paper shows is sonographic evidence that the structure of chimp and gorilla and orang, quote, laughter has the same sonic building blocks that human laughter does, more similarities there. So we've got this laughter-like stuff when other apes are playing, wrestling, tickling. What is play about in some other species? Exactly the same as in us. Play is when you feel safe enough in your environment that you are willing to give away some control, some predictability. Surprise me. Surprise me with something unexpected that will cause me to be delighted and will feel playful and perhaps cause me to laugh. Let's look at this clip showing what laughter looks like in an orangutan when it is caught by surprise with something in an otherwise safe setting. If that don't look like laughter at a lame magic trick, I ask you, what is that? Yeah, it sure looks like what we're doing when we're laughing. Probably the most important similarity is just as we like to laugh, we like to be put in circumstances that will evoke laughter, other apes do as well. One of the greatest summers of my life was after freshman year in college. I spent the summer working on one of those chimpanzee American Sign Language projects, which turned out to be a massive disappointment and way overblown and overinterpreted and anthropomorphized. But nonetheless, I got to spend the summer hanging out with this chimp named Nim, who was very young at the time and was just learning the first few signs. And he had maybe 3 or 4 of them. And like every chimp in every one of these sign language experiments, some of the first words he learned, some of his first sentences were tickle me. Technically, in ASL, this is actually scratch me, but tickle me. Tickle me. We spent all day long where I would tickle him and he would fall over and laugh and gasp for his breath and say, I haven't laughed like this since, and then roll up and tickle me again. This is how I spent the summer. It was heavenly. And probably the first conversation in American Sign Language between members of another species revolved around this as well. One of the other sign language projects was training 2 chimps in the rudiments of ASL. And one day they had their first conversation. They were sitting next to each other. And the first one says, tickle me. And the second one says, tickle me. And the first one says, tickle, tickle me. And then the second one says, tickle, tickle, and they go at it for a while, and then in a huff, they stand up and walk away. So just like in humans, these other species like to be in circumstances that will cause them to laugh. Now, amazingly, you also see rats laughing, rats giggling when you tickle a rat along the side, or when a rat that one rat likes grooms it along the side, the rat giggles. We can't hear it. It is ultrasonic. It is at 50 kilohertz per second, which is way higher than anything we can hear. But rats want to be tickled as well. How do you know? Put them in what is called an operancy. They will press a lever in order to have the grad student lean over and tickle them. They like it, just like the chimps, just like humans. This is remarkably similar. What we have here is a contrast between two terms in the business, whether something is homologous or analogous. Homologous, two organisms share a trait and their ancestors shared it, and their ancestors, all the way back to their last common ancestor, had that trait. That represents a homology. For example, all the mammals out there are tetrapods. They've got some version of four limbs, two arms, two legs, some version of that. And you look at the earliest mammalian fossils, and tetrapodishness goes all the way back. That is homologous in mammals. In contrast, you look at the fact that a bat has a wing, a bird has a wing, humans with the right contraptions could come together with something wingish. Are all of those homologous? Obviously not. They are not all descended from a single common ancestor that had wings. So we've got the difference between something that is homologous or analogous. And thus we ask, is laughter in these other apes homologous or analogous to us? And I think what we see is the anatomy of the larynx, the structure of the quote laughing, the facial expressions, the social context, the fact that it is pleasurable and reinforcing in all these cases makes me pretty convinced that this is a homology, that this is something deeply shared among apes. Apes have been laughing for a long, long time, and who knows what rats have been up to during that period. One final study showing the effects of animal laughter, which is extraordinary, which was a study done with rats by Rafał Regula, a Polish scientist who led a team looking at optimism and pessimism in rats. Okay, here's what you do. You train a rat. When you play a high-pitched tone, the rat's supposed to run over and press a button, and it gets a reward. Now, if instead you play a low-pitched tone, the rat's supposed to run over to a different lever and press it, and by doing so, it avoids the shock. This tone, good news, if I act, I'm going to get a reward. This tone, bad news, I have to proactively prevent a shock from occurring. So the animal learns this contrast just fine, totally accurate. Now, play a tone about halfway in between. And does a rat hear it as half full or half empty? Halfway in between, does the rat interpret it optimistically, saying, oh, that must be one of the good tones. I'm going to go hit that button. Or is it a depressive, ruminating, negative rat who says, oh, I'm not, that sounds like the negative one. And you see differences in strains of rats as to whether they go in this direction or not, differences in early experience, childhood adversity, all sorts of stuff like that. So in this marvelous, wonderful study, they showed that you take some rats, and if the experimental group gets tickled a lot, the rats become more optimistic. So I leave you all with that note.
There we go. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.